Now to talk about chromosomes, a non-membranous organelle, our first non-membranous organelle to talk about. We know that chromosomes are made primarily of DNA, and DNA stands for deoxyribonucleic acid. DNA, deoxyribonucleic acid. Now the DNA is made up of strands that are made of nucleotide units. And the nucleotide units, let's see if I can get this cleared up here. Nucleotide units consist of a sugar, a phosphate, and a base. And here's the bases are in these colors here: thymine, adenine, cytosine, guanine. So this thymine, a sugar represented by the red S, and then a phosphate by the P with the circle around it, that makes up a single nucleotide unit. So this is a nucleotide. And it binds across to the other side of the double helix to another nucleotide on this side. So there's a second nucleotide. Notice that the phosphates are opposite one another here. One's down below the sugar, or the other one's up above the sugar. So anyway, we have this binding between these nucleotide bases in here, the A and the T in this case. A represents a nucleotide base we know as adenine. T is thymine. C is cytosine. And G for guanine. So those are your four nucleotide bases. Adenine and thymine are always together, bind together, across from side to side, and cytosine and guanine are always paired in the structure. So anyhow, that's how we make our double ladder structure of DNA with nucleotide bases that bind across between the nucleotides here. So it's sugar, phosphate, then sugar and phosphate, and sugar and phosphate making up the sides of the ladder, and then in between the nucleotide bases, adenine and thymine binding with each other, and then cytosine and guanine binding with each other in the middle here. So that's your basic structure, your internal structure of a strand of that DNA molecule, deoxyribonucleic acids. So now we know what a nucleotide is. We know what the bases are, these four right here, nucleotide bases. And we know a little bit about the orientation of those bases and the nucleotides within a double helix strand of DNA. So this ladder here is twisted in such a way that it looks something like this image above there. So that's that double helix structure. So that's where you'd see all the binding of adenines and thymines and guanine. And anyway, you can't see much of this detail anyhow, so I don't need to go into great detail on that structure. But you can kind of see how it twists around to form that double helix. Next, this strand forms a strand that wraps around groups of proteins we call histone proteins. The histones, special proteins that help with the aggregation and condensation of the chromatin material, the chromosomes. So that in turn is twisted and on itself and again, and so on, until you get a really bunched up bunch of DNA and histone proteins and other proteins that help with the aggregation of the condensed chromosome, and you end up with the final condensed form of the chromosome to look something like this, which is what we see when we talk about cell division and talk about separation of chromosomes in the mitotic process. So a couple of terms we need to know here. First of all, in the dispersed form, the chromatin material, the sequence of nucleic acid bases, are useful for different processes of transcription and in replication of the DNA. So to be functional, it has to be in the dispersed form we know as euchromatin. Here's where it's spread out and along its length like this. When it's gathered up and bunched together, this right here is what we call heterochromatin. Euchromatin, and U usually refers to true. So this is true chromatin, is in its spread out form, so it's useful. It's doing its intended purpose for coding for the things we need to do, and then at times we need to stop that process of coding for more RNAs, DNA copies, or whatever, and get ready for the cell division process, and it bunches up to form this heterochromatin. It's more condensed form of the chromatin. So this is the less condensed, more spread out form of chromatin, euchromatin, and this is the all condensed and crunched up, not being used at the time for transcription or being replicated at this stage. It's getting ready to form this chromosome structure that we're familiar with. So that's the basic structure of chromatin in a chromosome. So now just to get some notes on the board, we know that chromosomes are long strands of the DNA double helix and their associated chromosomal proteins. That's what a chromosome is made of, the constituent elements. And we know that it exists as heterochromatin, a very condensed form of chromatin where it's all bunched up, forming the chromosome, or that chromosome can spread out, where it's no longer bound and twisted and folded, where it makes up what we call euchromatin, that non-condensed form of chromatin in the cell. Now for functions of the chromosome's functions, first of all, contains gene sequences that code for proteins and RNA molecules. All the proteins of the body are assembled according to a certain sequence of necessary sequence that makes them specialized and specific for each function of every protein out there. And RNA molecules also have to be put together in a particular way according to a certain code. The RNA, of course, first is copied and sent out into the cytoplasm for the construction of the protein. So it's an intermediate. And there are some RNA molecules that are, of course, there just to be RNA molecules, whether they're part of a ribosome or part of the transfer RNA mechanism. Anyway, there are special RNA molecules that have to be structured in a certain way according to the genetic code. So it contains gene sequences that code for proteins and RNA molecules. Next, replication. Replication of the genetic code before cell division. So you have to make copies of the genes before your cell divides so that each cell will have that genetic information. So they have to make copies, the replication process. And then third, inheritance, passing the genetic code on to the offspring. Pretty obvious there, okay? Those are functions of DNA or the chromosome. Now, also inside of the nucleus, along with those chromosomes, we have a structure called a nucleolus. Those are the main two features we find in the nucleus, chromosomes and nucleolus. The nucleolus is a small, dense area of proteins and RNA. So if we look at this was a cell over here, it would have a nucleus inside, and in there there'd be a dense little area, and then there's not much under a light microscope you can make out other than that there's a small, dense, usually dark staining area there inside the nucleus. That is composed of RNA molecules, special RNA molecules, that are vital to the production of ribosomes and certain proteins that are involved in the production or assembly of ribosomes. So, the nucleolus is a condensed region of special RNAs and proteins for the production and assembly of ribosomal subunits. Ribosomal subunits. So that's the overall structure of the thing, what makes it up, just this aggregation of proteins and RNA. And we've already identified the function as production of ribosomal subunits. We'll talk about ribosomes next. Okay, now for the ribosome. This is a ribosome here made of two subunits. There's a large subunit and a small subunit. These are made separately and then put together later. They are vital to the production of proteins. Now, these are made of a combination of both RNA and proteins. Each of them has several RNA components and several protein components in each one. But the two of them come together as a complete ribosome, so there's a whole ribosome, and they are responsible for stringing together amino acids in a sequence. So all of these different colored things along here, like colored beads, this is an amino acid chain. The sequence of amino acids is determined by a messenger RNA molecule, which I don't have in my drawing here, but anyway, it lies in here and forms a sequence code for the assembly of those amino acids. Up here I have a couple of transfer RNA molecules. We call them tRNA. They transfer amino acids to the ribosome, so they're going to be coming in here and adding more amino acids. There's a single amino acid attached to a transfer RNA. So it's bringing it in to be added to that in the correct sequence as coded for by a messenger RNA molecule. So here we have all of our RNA molecules kind of represented, though I don't have my transfer RNA molecule in there. Let's see if I were to do that in a different color. I might have the coded sequence of a messenger RNA, so that would be my messenger mRNA here. Ribosomes are made of the ribosomal RNA, and then there's transfer RNA. So anyway, transfer RNA bringing amino acids down here to the ribosome, and at the ribosome that code in the messenger RNA, that sequence of bases in the messenger RNA, will code for which amino acid goes in place next along this chain. And eventually this chain of amino acids will develop a three-dimensional structure that's vital to its function in the body. And there are thousands of different types coded for by thousands of different codes that originate, of course, in your DNA. So that's a ribosome. Now for our description, our written description, ribosomes are small, odd-shaped clumps composed of a small ribosomal subunit and a large ribosomal subunit. Each subunit is made of several RNA, ribosomal RNA segments, and several proteins. S on there, don't I? Segments. Several RNA segments and several proteins. Each is very specific to the structural composition of the overall subunit. And then those two subunits, large and small, come together to make up a complete ribosome. Then the function of the ribosome is the essential mechanism of protein assembly. At the ribosome, transfer RNA molecules bring amino acids to be added to a growing amino acid chain. So that's what the ribosome does. Now for an odd organelle that's only recently been included in many textbooks as an organelle within the cell, something called a proteasome. I've tried to draw one here, but it's described as a tiny barrel-shaped structure made of special protein complexes. Now the entire structure is probably not even visible under a light microscope. You'd have to go to an electron microscope probably to see the structure overall. But as this structure encounters proteins floating around within the cytoplasm or within the nuclear region of the cell, it attaches to those loose proteins and breaks them down into small fragments. It's kind of like the little wood chipper of the cell when it comes to proteins. So its function, digestion of loose proteins in the cytosol or nucleus, and digest them into short peptide chains, short chains of amino acids, or little peptide units in the cell. Then if it's going to be digested further, there are other enzymes within the cell circulating within the cytoplasm and nucleus that would break down those smaller units into their subsequent amino acid parts that could then be picked up by transfer RNAs and assembled into new peptide chains and proteins. So, little tiny barrel-shaped structure made completely of protein complexes, and like I say, it's kind of the wood chipper of proteins in the cell. Now for microfilaments. Microfilaments are made of subunits of a globular protein we know as actin. A-C-T-I-N, actin is a globular protein, and that actin molecule forms continuous strands that are like two sets of beads strung together. So there's one set of actin going through here, and then another wrapped around it right there. So this double strand of globular proteins of actin make up what we call microfilaments in the cell. As far as we know, there's only one kind of actin, and that comprises all of the microfilament structures we know in the cell. So we know that microfilaments are made of a twisted double strand of globular proteins. Those globular proteins are known as actin, that globular structure. So two strands, like twisted beads, strings of beads. Functions: they have primarily a cytoskeletal role in support of the outer membrane of a cell, beneath the cell membrane, kind of supporting that from beneath. They are also, we've already seen that they're in the core of a microvillus and a stereocilium. They make up this main body structure inside the cell beneath the membrane in those structures, microvilli and stereocilium. They're also involved in the sarcomere as the thin filament of the sarcomere unit. When we talk about skeletal muscle and smooth muscle and cardiac muscle, we'll look at that sarcomere unit, the elements that are involved in the contractile process, and the actin is an important part of that thin filament of the sarcomere. Now a word about intermediate filaments. Intermediate filaments. In the cell, the intermediate filaments are composed of sets of proteins that form these kind of twisted together pieces. So you'll get small pieces, chains of proteins to come together to make an initial component of the intermediate filament. Those pieces will then come together to make up strands of grouped or clumped proteins in kind of a middle stage, and then those come together with others to make up essentially a long cord of proteins in the cell we call the intermediate filaments. And unlike the actin type that we saw in the microfilaments, these of course get much thicker. Like instead of being a single thread, these form a cord relatively. So these are heavier, thicker filaments in the cell, these intermediate filaments. And instead of just being made possibly of one kind of protein, there are a lot of different kinds of intermediate filaments made of different kinds of proteins. Usually within an intermediate filament there's one primary protein type, but there are a lot of different types of intermediate filaments with different kinds of proteins that make each one of them up. So those are intermediate filaments. Okay, as we said, the structure of an intermediate filament is composed of protein strands which come together in other pairs or groups of four and then combine with other like groups to form a cord-like filament, like forming a rope. Several types made of different protein types are out there, so there are a lot of different kinds of intermediate filament, unlike the microfilaments which are only made of actin. Functions of intermediate filaments, there are many, and some of them I'm not familiar with. These I know about. Cytoskeletal helps make up that framework inside the cell, and often some of our attachment proteins will bind across to these intermediate filaments inside the cell to stabilize like a desmosome attachment from one epithelial cell to a neighboring epithelial cell. So they go through the membrane and then attach from the membrane to these intermediate filaments on the inside of the cell. Myosin functions, there are several kinds of myosin. We always think of the muscular myosin, so it's found in the sarcomere as the thick filament of the sarcomere, but in some cases myosin is just an attachment kind of protein. Other kinds of myosin are there to connect different parts of the cell inside. Formation of keratin. Keratin in your skin and your nails and your hair and so forth is primarily a intermediate filament kind of protein that makes up that keratin, that protein material of your hair, nails, and skin. And then finally the nuclear lamina, which we described the other day when we were talking about the nucleus and the formation of the nucleus. Nuclear membrane, inside the nuclear membrane. So we have that mesh, that kind of like a screen-like arrangement of intermediate filaments coming together there. So those are some of the functions, not all, but some of the functions we know of associated with intermediate filaments. Microtubules are made of subunits, just like as we saw in microfilaments and intermediate filaments. There are proteins that are the building blocks for these structures. And in the case of microtubules, it's a double protein structure made of two different kinds of what we call tubulin. This is called the alpha tubulin. And down here we have a beta tubulin. It makes up a tubulin dimer. These tubulin dimers bind together with neighboring tubulin dimers and they form a kind of tube, a circular arrangement here of these dimers where the alpha tubulin of one dimer will bind to the beta tubulin of the one above it. So you end up with this very organized arrangement of these proteins within the microtubule structure. And as they bind together to one another, they form a tube that kind of spirals downward along the length of the tube. So they kind of stair-step as they bind side by side to one another, and as you go around the spiral, they'll go around to where the next set is down below here. To get to the next step, there are 13 of the tubulin dimers that bind together in this circular shape of the tube in the microtubule. So I have 13 little blue dots in this circle, and say this one right here would be on the upper step, and as you go around this stair-step arrangement down to the lower level, the one right here would be just ready to go up underneath that top set of tubulin dimers. So you're going down, down, down as you spiral around the microtubule there. While there are 13 in the typical microtubule, as a single microtubule like this one right here, sometimes you'll find doublets of the microtubules, as we see in a cilium, in the structure of a cilium, around that 9+2 arrangement, the 9 pairs. When they form a pair like that, then the arrangement around here binds to that first set in such a way that there would be 10 here, then there would be 3 shared by the 2 microtubule structures. And then same sort of thing when you go to 3. So there's a single, there's a set of 2, and here's a set of 3. You'll find 3 tubules, microtubules together in the basal body portion of the cilium down there. 9 sets of 3. In the upper portion of the axoneme, you'll find 9 sets of 2, and in the middle of the cilium, you'll find 2 separate individual single microtubules in there. So that's the basic structure of microtubules. tubulin dimers made of alpha and beta units. They bind together side by side to form this spiral of 13 individual tubulin units around one circle of the tubule, and they stair-step downward so that they spiral downward as you go along the tube. So we just described the microtubule shape. So they were made, they're tubules, kind of like a straw, with walls made of tubulin dimers arranged in 13 columns in a spiral formation around an empty central space. Just as we described. I don't know if that written description quite does it justice, but with the description and drawings that we've seen, I think you understand the overall shape of a microtubule. Microtubule functions are fairly diverse. They make up part of the cytoskeleton, and in that cytoskeleton they can serve as kind of an intracellular transport structure. There are molecules that will attach to the tubules and something that needs to be transferred from one place to another inside the cell. So there's a mechanism whereby a carrier molecule will move objects around within the cell along these microtubules. Also, centrioles and spindle formation. Centrioles found just outside the nucleus, bundles of microtubules arranged in here, and then when the cell gets ready to divide, those help to form the spindles, and chromosomes will move along those spindles toward the ends of the cell to separate the chromatin during cell division. So those are functions there. Ciliary and flagellar mechanisms. We've already talked about cilia and the arrangement of the microtubules within a cilium, so we know their role there. Flagella, really only a sperm is the only thing really that has the flagellum, but it has basically the same kind of arrangement we see in the cilium for movement of the flagellar structure. So those are some functions of microtubules in the cell.